So, good afternoon, everyone. Welcome to the afternoon session. Hopefully, we are going to keep you awake after lunch with a really exciting session with an incredible panel of international speakers that includes two perspectives from the community. So just a reminder that what's set up here is not necessarily a reflection of the CF Foundation's opinions. So just as a reminder for what was in the program, our learning objectives for today are to describe the pipeline of nucleotide-based therapies, also sometimes called nucleic acid-based therapies with the same abbreviation, and the path to ensuring that CFTR-directed therapies will benefit all people with CF, including those who have a high unmet need and are from diverse communities. Also to review the clinical trial designs and recruitment approaches that are necessary to support the development of these therapies. These aren't modulator trials. We're sometimes going to do first in human with these studies. So there's additional regulatory considerations, including across diverse populations and people who may not have participated ever in a trial before. Utilizing knowledge that's developed in other disease settings and based on current regulatory guidelines, which may change as we go through this whole process together, understand the requirements for long-term safety and follow-up of nucleic acid-based therapies, anticipate progression of these therapies from adult to pediatric populations, and then opportunities and challenges with the adoption of these therapies across diverse populations. So again, I'm just floored that we got all of these amazing people to speak today, so thank you for showing up to hear them. And with that, I will take it over to our first two speakers. So we do have two speakers for several of the sessions. So Steph Hansen is a patient advocate and speaker from South Texas. She is currently serving as the 2024 National Peer-to-Peer Ambassador for the CF Foundation and serves on the Adult Advisory Council. Then Professor Scott Bell, who I'm sure needs no introduction to most of you, is the Chief Executive of the Translational Research Institute and a Senior Physician of the Adult CF Center at the Prince Charles Hospital in Brisbane, Australia. And he has obviously published numerous publications and really led some of the development of guidelines that we read today and use in our patients. So, do you want to come up?
I promised myself I wouldn't hit the microphone as I sat down. There we are. Blame jet lag. So welcome everyone, and I'm going to pose a few questions at the start of this session to Steph. The first one is, Steph, as we just heard, you have CF. Can you tell us a bit about your story?
Yeah, I'm 35 years old, living with cystic fibrosis that's caused by two nonsense mutations. I'm from San Antonio, Texas.
And having two nonsense mutations means that you've not been able to access the modulator drugs as we currently have. What impact does that have had on you?
It's been emotional. I've felt really lonely and at times left behind, especially with all of the celebration around modulator therapy and then the disparity that's been created between myself and those who can access these therapies. I'm so sorry, I hit something. But my heart has sort of failed to understand why all of these people sort of got life jackets or this life-saving drug while I have been left to just tread water, especially as my disease continues to progress. And that has led to a pretty incredible sense of desperation. I want to do anything I can to change that, and that's what led me to research.
Steph, obviously you've considered involvement and participated in gene therapy-based treatments or gene-based treatments. What concerns did you have leading up to deciding whether you would participate or not?
Well, I heard about some stuff that happened in the 90s, and that is definitely a concern. But, I also just have health concerns of my own. If I were to have an adverse reaction to one of these new drugs, I was really concerned about the potential treatment for that and what that would look like. antibiotic treatment has become very, very difficult for me. So I had to weigh the pros and cons and decide if it was worth the risk to my health in order to participate in a trial like this. I think another factor for me was feasibility. I don't live near a large research center, so I knew I'd have to travel. And I wanted to ensure that as many of my needs were met on the front end. I think that even with reimbursements, the out-of-pocket costs can add up, especially with longer visits for these trials. And as somebody who doesn't work, my health is my full-time job. That was definitely a concern, and I had to lean on family for support to get me through this trial. So that was a big factor for me. Additionally, while this isn't specific to this type of research, one thing that I always consider is I am always a little bit apprehensive to work with new care teams. Because of my openly queer identity, I am never sure if I'm going to be treated differently or if my care is going to look different because of that. I actively avoid research opportunities in certain parts of the country because of how I've been treated in those parts of the country. So ensuring my safety, both in the clinical setting, but outside of the clinical setting, as I travel was at the forefront of my mind when choosing to travel for research.
Thanks for sharing that, Steph. Why did you decide to go ahead?
I'm sorry?
Why did you decide to go ahead with the studies?
Well, that desperation I talked about, it fueled me. The research team that I met upon screening, they were really, really passionate and very excited about what they were doing. And I realized that made me feel less alone. Again, this was my first time participating in research, and to feel that from the research team and knowing like, hey, there's all of these people who are trying to create better treatment options for me, that was so exciting and it meant a lot. And I realized that taking action in the form of participation, it counteracted the feelings of powerlessness that I had felt as somebody who can't take modulators. So I also realized then while screening that I would reach out to friends I thought might be interested or might be able to take part in these types of trials, and all of them, not a single one of them were able to participate due to not meeting eligibility criteria, health status, or the time investment needed. It's a lot. So at that point, I felt almost a sense of responsibility. And I was like, well, if I'm not going to do it, who's going to do it? So, I certainly want a healthier future for myself, but I also want a healthier future for those people who really need these drugs even more than I do, and the kiddos who come after me because they can't participate in these kinds of trials either. So it was ultimately a sense of responsibility.
Thank you, Steph. Join with me in thanking Steph. You'll hear more from her a bit later in the afternoon.
And as Scott heads up to give us his talk, I just wanted to point out that we are going to save all of the questions until the end. So if you'd like to put one in on the app, great. We'll keep an eye on those, but everybody will come up at the end for a panel.
Well, thank you very much, Jen and Nicole, and to the organisers for inviting me to speak today. And I guess it's a bit of a letdown after hearing Steph speak. What can we say about the patient journey that hasn't already been presented and will be presented later in this symposium? Here are my disclosures. I wanted to add two to this. Firstly, you're going to hear a lot more about the technical aspects of nucleic acid-based therapies in the first plenary. So don't expect to come to me as the expert. Save those questions to a bit later in the afternoon. And secondly, there were a lot of different potential therapies that I could have talked about today, and I've chosen strategically a few of those because of the limitations in time. So let's think a little bit about the current context. Who are the patients that are not currently able to access CFTR modulators? They're those with a non-druggable variant, and there are certain populations that are overrepresented. These are the current CF Foundation patient registry data, including patients who have had transplant, and we won't talk about those today. Of the 33,000, 3/4 of the population are able to access one of the different CFTR modulators, the vast majority ETI. The orange segment of the pie represents those that are currently ineligible, either on the basis of age, that the specific drugs are not currently registered for use in populations, usually young children, or secondly have a CFTR variant which is non-responsive. That represents about 8% of the CF population in the US. In the blue segment of the pie, you see a combination of people who either have decided not to currently have CFTR modulators or have had an adverse event. And we'll talk a little bit more about that during the course of the symposium. But we're really focused on that group of patients as the unmet need at the moment. Of course, we need to think about the more global perspective, and there are many tens of thousands of people around the world with cystic fibrosis that for various reasons, including diagnosis and availability of the modulators, are not able to access drugs. So let's talk a little bit about the purpose of nucleic acid-based therapies: to reprogram the CF cell to make functional CFTR protein by providing the correct instructions. I'm going to talk about 3 of these groups of therapies: antisense oligonucleotides, mRNA therapies, and DNA-based therapies. I'm going to focus largely on gene therapy or gene replacement therapy. If you wish to go and find more about gene editing-based therapies, there are at least a dozen posters on this throughout the hall downstairs, and this field is obviously changing very rapidly. Let's focus a bit on antisense oligonucleotides. Currently, they aim to focus on specific variants which are currently not druggable with modulator drugs. The 2 specific classes of variants are nonsense variants, such as the W1282X mutation, and splicing variants, such as the 3849 plus 10 kb C greater than T variant. But there are potentially other types of variants where increased translation through to protein production suggests that antisense oligonucleotides may be useful in the future. They're able to be inhaled to the site of action, and a carrier may be present, but not universally so. So in other words, the antisense oligonucleotide can be nebulized in its native form. Let's talk a little bit about mRNA and DNA therapies. So both are variant agnostic. mRNA therapies are generally packaged in lipid nanoparticles and aim to take normal CFTR mRNA and deliver it to the cytoplasm of the cell, and then subsequently normal CFTR protein is produced and expressed on the epithelium. I note, and we'll come back to this, that there have been some challenges with early trials of at least one form of mRNA inhaled therapies. DNA replacement or gene replacement therapy has had a history going back 30 years and has included viral and non-viral vectors or carriers. Classic gene therapy aims to correct the CFTR DNA by inserting it and subsequently transcribing the CFTR protein inside the cytoplasm of the cell. For the purposes of this talk today, I'm not going to cover gene editing. mRNA therapies are generally inhaled and are thought to, in the future, require repeated application. Follow-up is required and our understanding of off-target effects important, as I'll return to. For gene replacement therapy, the therapies are generally inhaled, and there is some potential, at least, for longevity of response. So the duration of effect is as yet unknown, but likely to be longer than mRNA therapies and antisense oligonucleotide therapies. So to summarise, redosing will be necessary, and at least for some forms of gene therapies, antibody development may preclude that. Early phase participation might prevent eligibility for future studies, and these approaches will potentially improve airway disease, but not necessarily impact on disease within other organs. I'm just not sure what's going on here with the lights. Keep going.
Yeah, go.
Yep. So let's talk about the program by SplySense, the Sply-84 antisense oligonucleotide therapy. This has been trialled in healthy volunteers and in a phase 1 clinical trial with people with CF, where abnormal splicing of the CFTR mRNA prevents functional CFTR. The antisense oligonucleotide helps normalise the splicing to enable fully functional CFTR. In the laboratory, Sply-84 restores CFTR activity and has a promising safety profile following inhalation. In a single ascending dose study in healthy volunteers, no serious adverse events or significant treatment-related adverse events were detected, suggesting a promising safety profile. Systemic exposure to Sply-84 was low and dose-dependent. A phase 2 study with people with CF is underway. This is a placebo-controlled, multiple ascending dose therapy in patients with the specific splicing mutation. The primary objective is to evaluate the safety and tolerability of multiple and ascending doses of Sply-84. And clearly it's important to look at the secondary objective, which is finding preliminary evidence of a lung function signal. I'd like now to turn to mRNA therapies, and I'm going to focus most of the discussion on the 3 particular products here. But I want to start by talking about Translate Bio and their inhaled CFTR mRNA product that underwent phase 1 and phase 2 clinical trials. The results of this study were published in the Journal of Cystic Fibrosis, led by Steve Rowe, and some important messages were noted. There were concerns about the safety profile, and some of which were significant. There was a suggestion of both systemic adverse events and also airway-based adverse events. In the phase 1 study, 2 of the 4 participants appeared to have a significant lung function response. However, in the phase 2 study, this was not replicated. And the development program has now been suspended. There's some important lessons for the other producers of mRNA products here in terms of off-target impacts and systemic side effects. Vertex, in collaboration with Moderna, are developing an investigational mRNA therapeutic, which is being delivered through a liquid nanoparticle delivery directly to the lung. A multiple ascending dose study is currently underway in adults with cystic fibrosis. It's delivered through a vibrating mesh nebulizer, and the multiple ascending dose arm of the study is interesting in that the first arm involves the investigational product, and the second arm also has the supplement of ivacaftor. Patients participating in this study need to have CFTR variants on both alleles that are non-responsive to modulator drugs. The primary objectives are safety, but also evidence of spirometry improvement in the secondary endpoints. And as highlighted with the previous slide, evidence of immune response and systemic evidence of the therapy in blood is being sought. The next mRNA therapy that I'm going to cover is the Arcturus Luna ARC-032 mRNA replacement program. This mRNA is encased in a proprietary Luna liquid nanoparticle for inhalation. And the investigational product has been demonstrated to transfect airway epithelial cells both in vitro and in vivo, and has shown evidence of restoration of chloride transport in human bronchial epithelial cells. Using the G551D CF ferret model, a single dose of Luna mRNA demonstrated significant mucociliary improvements in mucociliary clearance, at least, if not better, than that seen with Kalydeco. Both the phase 1 Part 1 and Part 2 trials have now been completed. In the phase 1 Part 2 study, which involved 7 CF adults, patients with any CFTR variant were allowed to participate in the study, and continuing CFTR modulators was deemed reasonable as this was a safety-focused study. In the past several weeks, the FDA has cleared moving forward this program into a phase 2 study, which will also be a multiple ascending dose study in adults with CF, and it is envisaged this study will start very shortly. The objectives include safety and tolerability, but also efficacy from a lung function point of view with repeated doses of Luna CF. And this will be in patients who are adults. The final mRNA therapy that I'm going to talk about is Recode 2100. This slide demonstrates the in vitro impact of this preparation of mRNA on human bronchial epithelial cells. On the left, homozygous F508 cells, and on the right, heterozygote minimal function HBEs, with a significant efficacy seen of the order of 75% in the homozygote cells of Trikafta and greater than 100% in the het min function cells. As seen previously with the earlier study by Arcturus, improvements in mucociliary clearance have been demonstrated of the order of that seen with up to 2 weeks of ivacaftor. The phase 1A single ascending dose study is currently underway, and the phase 1B multiple ascending global study will commence in the US, the UK, and Europe shortly, and will be enrolling patients who are not taking CFTR modulators, both those that are currently ineligible or intolerant of currently available therapies. Finally, I'd like to talk about gene therapy, and I've specifically decided to talk about 2 programs, the 4DMT program and the Boehringer Lenticlaire TM program. The former involves a vector using the adeno-associated virus, and the phase 1/2 program is ongoing. The Boehringer-Ingelheim program uses a lentivirus vector and is envisaged to start late 24 or early 25. This slide demonstrates this journey over the past 30 years of gene therapy trials involving a number of different viruses, adenovirus, adeno-associated viruses, and lentiviruses, as well as non-viral vectors of liposomes and lipid nanoparticles, all at an early stage of development in terms of clinical trial programs. The 4D710 design involves a lung-specific capsid which encapsulates a codon-optimized CFTR gene and a CMV173 promoter, which has been demonstrated to lead to effective mucus penetration, efficient transgene expression, and resistance to pre-existing antibodies. This is an interesting ongoing study in that it's a dose de-escalation study. Bronchoscopy is undertaken at 4 weeks, and patients are studied for up to 2 years with longer-term follow-up schedules. It is being undertaken in adults who are ineligible for CFTR modulator therapies with moderate to mild CF lung disease. And the early studies are promising in terms of gene expression, CFTR protein expression, and localization in the apical regions of bronchial airways. Finally, the LentiClaire program, which aims to treat genetically ineligible patients who are genetically ineligible for CFTR modulators. Pretty standard inclusion and exclusion criteria. I draw your attention to the CFTR modulator requirement and also the importance of being naive to gene therapies in the previous 6 to 24 months. On this right-hand side of the panel, you see the primary endpoints and the clinical trial design, which will clearly be very complex and challenging to undertake. Patients participating in this study will be monitored according to the regulators for up to 15 years after administration of the investigational product. Finally, I'd like to just talk a little bit about the title, which was around investigator aspects of clinical trials. Many sites will be sought after and offered more than one trial, potentially simultaneously. On the other hand, some trials might only be available in a single or several sites nationally. And that's not just for nucleic acid-based therapies, for novel CFTR modulators and symptom-based therapies such as anti-inflammatories. Even more than in the past, site and clinic communication and prioritization is going to be so important. We may need to undertake endpoints which are new to centres. LCI is not available in all adult centres and many pediatric centres. Rolling out imaging and nasal epithelial studies will be new to centres. And for early phase studies, which may require inpatient facilities, will be challenging in many clinical settings. We'll see less patients meeting criteria. We'll have patients seeking participation, or will be seeking participants who are unfamiliar with clinical trials. And there's going to be less patients available to participate in many different studies. So Taylor Swift has become quite important in this country over the past few weeks, but I wanted to remind you of her recent statement that there's a lot going on at the moment, and I think that's relevant to this particular area. Companies, regulators, centres, participants and their families, and our biostatisticians are challenged. The key aspect of these drug development programs is providing options for those with non-druggable variants or those that are intolerant to currently available drugs. But we also need to think about the opportunity for better options for those who currently access standard of care, and also whether there might be potential value add on top of standard of care. I'd like also to thank the many people who participated in providing me with pre-publication slides, and look forward to discussing further. Thank you.
Thanks a lot for that overview, Scott. And just for those of you who came in a little bit later, we're going to save questions for the end. Please feel free to put them in through the app, but we'll have everyone come up at the end for a panel. So again, another speaker who everyone in this room, I think, understands, helps design all of these clinical trials. So Dr. Nicole Mayer-Hamlet is Professor of Pediatrics, an adjunct professor of biostatistics at the University of Washington. Of course, she is also the co-director of the CFTDN. Nicole?
All right, so challenge accepted. Thank you again to Steph Hansen and Dr. Bell for really setting the stage. As you've heard about the breadth of our NABT pipeline and the urgency and care that we have to progress this pipeline across our diverse CF community. Okay, so it is not uncommon that I look back on my talk title and want to change it slightly. The word considerations was giving me slight pause, as I realized that I wanted to emphasize how we are moving beyond simply considerations to strategy to move this entirely new class of therapies forward for CF, in some cases where strategy has never existed before. The number of cell and gene therapy trials has increased dramatically over the last decade, and we are anchored by emerging regulatory guidance that begins to tackle some of the complex trial design issues for these therapies. However, to move an entire pipeline forward through clinical development, it is not surprising that we are going to need our own set of CF-specific, disease-specific guidance and strategy to move more than one shot on goal forward through clinical development. So today I'm going to highlight some of this strategy, the key word here being evolving. And beginning with that which is needed to help us maximize recruitment opportunities across the pipeline with a trial design perspective in mind. We know that development populations for NABTs will be initially focused on the approximately 10+%, maybe as high as 15% of people with CF who are not candidates for modulators due to either genetic ineligibility or intolerance. In the United States, this translates to be about 1,100 adults when applying historic eligibility criteria. Now, the reason I focus on adults is that these trials will begin in adult populations before they translate down into a pediatric population. And our next speaker, Jane Davies, will be speaking about what this transition may look like. We know that very few have had prior research opportunities or experience itself, and nearly 1/3 of this population identify as BIPOC and or Hispanic. And thus, it is of no question that it is going to take significant outreach, trust building, education, and innovation to meet our recruitment goals for these trials. And I'm happy to say that our plenary this afternoon will begin to talk about some of the broader efforts that we have in terms of our community engagement and partnership to move these efforts forward. I want to spend our precious time today focusing in on one of the more specific NABT development issues that we will need to tackle, one that's very unique to this class of therapies and that we continue to highlight to our regulators whenever we get the opportunity. And that's the need to mitigate the potential for a diminishing participant pool over time. And this is driven by two key factors. The first being something that Dr. Bell just referred to in the context of redosing, but the potential limitations to multiple NABT exposures that an individual may have. This could lead to hesitancy and or in some cases maybe an inability to include participants in subsequent NABT trials who have had select prior NABT exposures. Okay, so looking at our pipeline and mapping this out, you can envision that mapping out all the different potential combinations could get quite complex in terms of the key factors that can inform that opportunity for re-enrollment, moving from an mRNA therapy to a viral vector-based therapy or from a viral vector-based therapy to another viral vector-based therapy. At a very high level, no matter what the combination is, the observed safety and the observed clinical response in that first trial is going to be incredibly informative for even, informing whether another shot on goal is needed for that participant. It's when moving from a viral vector-based therapy to another viral vector-based therapy that there are additional factors at play, such as the potential occurrence and impact of an immune response, or predominantly in the setting of integrating viral vectors, the potential for off-target effects that could confound safety and efficacy assessments, particularly for that second therapy. However, leaning on the word potential here, these are predominantly theoretical concerns with an unknown frequency of occurrence and impact on efficacy or safety. This is an evolving field. Thus, our overall strategy here needs to proceed with caution with these factors in mind, but at the same time of promoting and developing eligibility criteria for these trials that can maximize the opportunity for re-enrollment for somebody who has had a past NABT exposure. Now, the second factor that we have going on here are the regulatory required long-term safety studies. These could range anywhere from 2 to 15 years for an integrating viral vector-based therapy right now. And they will also complicate participation in subsequent investigational trials. So you can envision a scenario where someone enrolls in a first trial, they get rolled over into that long-term safety follow-up study, and we may want to consider, is there another shot on goal for that participant? And additionally, when's the optimal timing for that shot on goal in relation to perhaps their ongoing participation in a long-term safety study? So all of this points to the need for novel guidance to inform the need, eligibility, and timing for subsequent NABT trials. So this guidance is actually under development. We're working on this within our network. It's not quite ready for prime time. But what I can say is that this is an opportunity. We're actually ahead of the regulators here in thinking about this. And it's basically an opportunity for our clinical development strategy to keep pace with the incredible pipeline that's coming and evolving and advancing for our cell and gene-based therapies. Okay, so I'm going to get a little bit back into my lane. That was some scientific grace there in that portion. But let's talk about some lower-hanging fruit to maximize recruitment opportunities for this pipeline. A recent study from Dober et al. Basically mapped historic eligibility criteria onto their patient population. And they found that 26% of 31 modulator ineligible candidate trial participants had an FEV percent predicted greater than 90% as one of their common exclusion criteria. So this begs the question, where does that cap come from, and is that something that we could revisit, particularly as we're in this ultra-rare population? So let's take a data-driven approach to this. And we looked at the 6-month average change in FEV among adults for some of our other CFTR-directed therapies, ivacaftor and ETI. Among those with a baseline FEV of 90 to 100 receiving ivacaftor, they did have slightly attenuated but still clinically meaningful treatment response. When you pull those patients in to an overall treatment effect from everyone from 40 to 100, it just has a minimal effect on that overall treatment effect. On the left-hand side is the pooled treatment effect from those with the baseline 40 to 90. But you also see this other phenomenon going on, and that the confidence interval shrinks. Look at the impact of having more patients to estimate that treatment effect. You see that even more predominantly with ETI, for which there was no attenuation of treatment effect in this group. And yet, we're pulling in more patients, can get a more precise treatment effect estimate. So this does provide a potential additional pathway as we move forward, and particularly as we progress programs through clinical development to, reconsider that cap. And can we look at our eligibility criteria to see how we can make it the most inclusive for this population? Maximizing recruitment opportunities across the pipeline is incredibly important for us to conduct efficient and early phase trials to help establish proof of concept. These trials have to maintain a critical strategic balance. So our early phase trials, we're attempting to establish safety, biological activity, which is tricky for this class of therapies. They are inhaled therapies, and we do not have systemic biomarkers such as sweat chloride. Signals for clinical efficacy and dosing. We're trying to figure all of that out. At the same time, we need to limit overburdening a limited size study population and, as mentioned before, exposure to potentially ineffective doses that could prohibit future NABT eligibility. So here's the good news, is that we can strike this balance with carefully designed early phase trials. And our clinical trial networks, this is exactly what we review as these trials come in. And we have a number of different safeguards in place to help us do this. Especially when thinking about, as much as we want to establish proof of concept and look at every single dose that comes in, we don't necessarily have the resources to do that. So we need to really critically review the data and decide, okay, which dose cohort do we want to expand at the right time to really investigate proof of concept. And that's where we'll have scientific review committees in addition to our data safety monitoring boards during this phase of the trial. And it will lead to more amendments, but these amendments, in my opinion, are definitely part of the strategy. They are helping along the way to help maintain this balance as these programs progress. As we complete these studies, we will eventually be making decisions about a go or no-go decision to pivotal. And yes, it's very classic to want to see safety as we can, biological activity, but we need to use some extra discernment here, given the limited sized study population. So we will need to see signals for clinical efficacy. It may not be a statistically significant signal, given we're working with small patient numbers. And although we want that optimal dosing strategy, we do need to be a little bit realistic, and we're looking for a dosing strategy, to confidently move forward. And I want to point you to a symposium that we have tomorrow afternoon that's going to be diving into this a little bit deeper, going over sort of for our historic chronic therapies, how the dose response curve has been defined and how that may or may not apply now for this newer class of therapies. Okay, so as we move forward through clinical development, we expect that we will be progressing some, but perhaps not all, to later phase development. And it is not too early to begin to discuss the clinical trial designs for these later phase studies, and in particular, designs that will enable multiple potential shots on goal. And, as much as you want to step into the design, you got to stop and talk about, well, what's the hypothesized treatment effect for these therapies? So here we have the anticipated NABT response region drawn in relation to what we consider are more highly effective modulator therapies and more modestly or moderately effective modulator therapies. Basically kind of a 5 to 7% FEV effect range. More than moderate is, I would say, an unofficial regulatory term to keep in mind. That's going to come up later. Why is it in the middle here? Well, first of all, these are inhaled therapies. They're anticipated to be less efficient than systemic modulators for the lung. And also, we need a range for which we can be able to rationalize that the benefits of these therapies outweigh the anticipated risks for this set of therapies. So now we can start to look at the feasibility of our trial designs, given this effective range for the effect size. So in terms of FEV percent predicted, it would take about 30 per arm to detect a 7% difference. That's not huge, actually, when from the perspective of moving a single program forward. Now, for looking at CFTR, patient-reported symptoms, it's noteworthy that it would take larger sample sizes than FEV. And again, we expect the effect sizes for CFTR to be less than that for modulators, because these are therapies really targeting the lung and not capturing some of those other potential benefits that ancillary benefits that we see from modulators due to the extrapulmonary effects. And although we expect these therapies to impact exacerbations, the sample sizes within this population still would be infeasible. As we think about the regulatory package needed to progress these therapies, it's important to note that biomarkers and other clinical endpoints are expected to be extremely supportive, but not necessarily serve as surrogates for regulatory accepted clinical efficacy measures. And by surrogate, I mean a substitute for these measures. And this includes, LCI and MCC and CFTR expression. I want to spend a moment talking about surrogate endpoints. and get outside of the CF bubble for a minute, because there's been a lot of chatter about the use of surrogate endpoints, particularly for this class of therapies. There's a lot recent approvals, say in Duchenne's muscular dystrophy, that are basing approvals based on surrogate endpoints. I thought this was a very interesting slide because there are criteria out there being suggested for the use of gene expression as a surrogate endpoint. And just want to highlight a couple of these criteria. Gene expression is documented in the relevant tissue and intracellular locations, so localization, at levels expected to be clinically meaningful. And gene expression is documented to result in functional protein. So I'm not going to go into how this measures up against our current assays. I'm not the one to do this. But what I can say is that if you wanted to make this link, you would need more bridging data here. And in particular, you would have to demonstrate the sensitivity and the specificity of protein expression in terms of predicting clinical outcomes. This is possible, although a bit challenging because this data does rely on bronchoscopies, et cetera. So we have to be a bit realistic. If that were achievable, perhaps, then it may put us on a path to being able to access an accelerated approval pathway down the road that would rely on surrogate endpoints. But we are not there yet. And thus, we would like to focus on other pathways to streamline development, such as the use of innovative clinical trial designs. Many of the cell and gene therapy trials are single-arm trials. This does not yet capture our current pipeline. And in addition, there's a lot of regulatory buzz about the use of external controls to help support a regulatory package. So our idea here is to perhaps streamline our trials with the availability of external control data that could help augment or perhaps even replace the need for a placebo control arm, thus streamlining the size of a trial and letting more trials progress through development. The caveat here is that these trials are only really appropriate under a more than moderate effect size assumption. Well, good news is that is actually the assumption that we are working with in this scenario. So we are actually building a sponsor-agnostic external control strategy that would support this approach. Not only do we have, perhaps registry data and past clinical trial data, but we have launched a study, REACH, which I would like to highlight as being led by Dr. Deepika Polanini. Please refer to poster 444, and you will hear a lot more about it in the plenary and throughout the conference. It is just launched, and it is enrolling now. And this study is really meant to be a fit-for-purpose study to collect research quality control data to add to this strategy. In addition, it is the mechanism for us to engage our diverse population in research, in a study that is not actually, introducing a therapy at this point in time. And so we're thrilled to launch this study, and we also have a relationship now with the Critical Path Institute, who has a novel data sharing platform that we envision can be used to share data with our industry sponsors. So I'll just end by saying that it's all strategy all the time, that this is just a snippet of some of the work that we are doing behind the scenes. And we have a home now on the CFF website. There are at least 2 different position papers here now that you can access that lays out some of this more strategy in more detail. And if you don't get a chance to capture this QR code, I hear it is also at the CFF booth. And lastly, I just want to end by thanking, there's a critical core team that's thinking about this clinical development strategy every day, and really highlight my key colleagues and partners, Dr. J.P. Clancy and Joe Paluski. Thank you.
Thanks very much, Nicole. And as our next 2 speakers join us up here, I just wanted to highlight one thing. I appreciate you modeling the use of the acronym BIPOC, Black, Indigenous, and People of Color. I think that most of us who fall in that category do not consider ourselves, quote, non-white. So I'd encourage all of you to consider that as you're creating your slides and your documents. So thanks for doing that. I think in the UK, you guys use BAME. At any rate, our next 2 speakers are really going to be great, I think, as well. So Rena Barrow is someone who has 2 sons with cystic fibrosis, but she also is a pharmacist for CVS and CEO and founder of 2 Salty Oaks. And then Professor Jane Davies, again, we probably don't need to introduce her, but she is a professor of pediatric respirology and experimental medicine at the National Health, Heart and Lung Institute, Imperial College of London, a National Institute of Health Research Senior Investigator, and a Fellow of the Academy of Medical Sciences. She is currently also the President of the European CF Society and a role model for many, many investigators, but particularly young women in science. So thank you both.
Good afternoon, everyone. Thank you so much for being here. Thank you to all the speakers here, and a special thanks to Dr. Jennifer Taylor-Kusar for inviting me. I'm so excited to be here today to share a little bit of my story and from a parental perspective on gene therapy and clinical trials. My name is Rena. Well, I came in from Atlanta. It is raining there right now. We're getting hit by the hurricane. My name is Rena Barrow Wells. I'm a wife and mother of 4, 2 boys and 2 girls. Both of my boys, ages 23 and 4, both have cystic fibrosis. They were diagnosed late. In addition to advocating for their health, I've spent the past 17 years working in the pharmaceutical industry, both retail and hospital pharmacy. And currently, I'm a senior pharmacy benefit manager for a large corporation. Do not stone me all at once. Just kidding. My sons, they have participated in research trials, and I have my notes with me so I can stay on task today. I'm on a time schedule here, and I want to be able to land my plane. So my sons have participated in research trials. The primary concern has always been trust, which has been historically damaged within the Black and African American healthcare community. The Tuskegee experiment and other instances of mistreatment of BIPOC communities, coupled with our own experiences with late diagnosis, has made us, me and my family, very wary of research. However, our personal journey with cystic fibrosis, from seeking a modulator to finally receiving one, has led us to believe that research may be our best hope for optimal health and a better future for younger patients, especially my 4-year-old and all of the patients that come behind him. I have 2 concerns. Well, I would say they're pre-concerns and also post-concerns. My pre-concerns were about the clarity of information sharing and potential risk associated with the trials. I wanted to be informed of all risk, both minor and major, and to understand any potential biases or nuances that might arise during the research process. To rebuild trust, I believe that using clear and understandable language or layman's terms is essential. Open communication and transparency are key to fostering a positive relationship between patients and also researchers. My post concerns, one of the main concerns is affordability of gene therapy once it becomes available. So I would like to ensure that those who actually qualify for this treatment have equitable access to it. Regarding gene therapy trials, I would definitely consider discussing participation with my children, especially if the potential benefits significantly outweigh the risk and could improve their overall health. When evaluating the feasibility of enrolling in such trials, I would carefully consider factors beyond the potential benefits for the broader CF community. For example, as a parent, I would need to assess how participation would fit into our existing schedules for standard CF treatments in care. Ultimately, our decision would be based on a comprehensive evaluation of the potential risk and benefits, taking into account our children's individual needs and also circumstances. Thank you.
Thanks, Rena.
Thank you very much, Rena. That was really powerful. And thank you to both of you for setting up this symposium in a slightly atypical fashion. I think it's really, really enhanced having the voice of you, Steph. Thank you so much for what you said, and really brings the relevance of it very much more to us than just a talking shop where we are, conversing as professionals. It's so important. So I've been given a talk in 2 halves, and I will come on a little later to talk about the topic of pediatrics in trials, as Rena was talking about. But first of all, a little bit about thinking of the long-term follow-up issues. So here is the disclaimer. I do have one other disclaimer personally, and that is that I don't have one of these. So some of what I'm going to say could turn out to be totally wrong. I'm trying to look forward and think about how things are going. I do have a number of relationships with pharmaceutical companies working in this space, and anything that could be construed as an objective opinion is my own in what I say today. So Nicole has already touched on this a little, and I won't labor the point, but to think about why do some of these studies attract such long-term follow-up periods? Why does that happen, and how should we do it? So I think it's important that we acknowledge this is a relatively new field. We're learning on the go, if you like, and even those areas of gene therapy which have been around for longer, they're also learning on the go. There is undoubtedly the potential for long-term issues, and some of those have become reality from some of the trials in other disease areas, and there is theoretical plausibility for other issues, even if those risks are considered low on the balance of all the information that we have available to us. I think it's fair to say that the area of focus in this regard is mainly on the viral approaches as opposed to some of the non-viral approaches. So these are some of the considerations by the European Medicines Agency, and they will be very similar to those considerations by other regulators. So they'll be wanting to think about whether or not the gene therapy vector that you're using has the potential for integration, and whether or not that integration could lead inadvertently to oncogenesis. Also, whether a vector or a gene has the capacity for latency or reactivation. Whether or not some of the reactivation could actually complement a naturally acquired virus and lead to a new virus entity or an infection, and also what the persistent characteristics and the long-term effects are. The persistence of the gene product and the unintended biodistribution is something that they also focus on, and replication competence. So this is quite a useful guidance document, and there are a number of evolving guidance areas coming from there. And one of the things that people think about is how long do we have to monitor and what do we monitor? Thank you to Nicole for passing this slide on to me. I understand this is something that's been pulled together as part of the initiative that you've been undertaking through the TDN-CREC. And there's a lot of information on this, but I'll perhaps just go straight to the long-term follow-up periods that have been postulated. And you'll see that there are very big differences in those periods depending on what the actual product is and whether that is a viral or a non-viral, and whether in particular for a viral product it's an integrating product. So in terms of the EMA's view on that, vectors which can integrate or have the potential for latency followed by reactivation, they mandate a monitoring plan which includes clinical history and sample testing at multiple time points. Those time points become wider apart as time goes on past the dosing. And if any of those post-treatment samples are positive, then that period of monitoring has to be extended. the data of special interest that they would be asking people to look out for, of course, mortality, but development of any new or recurrent cancer. And I think it's important to say that in many contexts of genetic therapies, those cancers might not be as easy to diagnose as they have, for example, in the blood-borne cancers that came out of some of the severe combined immune deficiency patients, where it's pretty easy to draw blood and to see that some of those cells have transformed. So it may involve imaging. One of the things I think it's important to say is that it is possible with modern technology to have a look at causality in terms of integration of a genetic product and actually to show whether or not it has been involved in the formation of that cancer. And I think that's something that I'll come back to later that's quite important. Immunogenicity was mentioned by Nicole, and then further safety endpoints, many of those on this slide, not really of relevance to us. They also draw attention to participation in other clinical studies. So that's something for which the door appears to be open from a regulatory perspective, which I think is very important. And then in other disease contexts, there may be possibilities of interactions with other treatments that people are receiving, for example, radiotherapy and cytotoxic agents. I think, fortunately, we're not really in that group there. One other bit that I found within one of the guidelines of the MA, which I thought was quite attractive, was that they particularly mentioned the remote conduct of follow-up activities and the use of digital tools. And I think if we're thinking about being creative here, this is one of the things that we should at least factor in. So in terms of long-term follow-up, the challenges for the CF community, I'm going to reiterate some of what Nicola said, but I think it is really important. Steph mentioned earlier that one of the things that influenced her about taking part in a trial was the distance traveled. Many of us are now setting up trial sites which have a hub and spoke facility so that we have smaller numbers of very expert trial sites and people travel to them. Now that's quite inconvenient, for example, for a 6-month trial. I don't know how long yours was. If you've got to then be doing that and going back to that site for a long period of follow-up as well, then that really becomes exponentially more inconvenient, and I think that's something that's important. People obviously have lives, they want to move, they sometimes want to move country. I think people getting lost to follow-up in CF is relatively unusual, but if people move to a different country, for example, that could be quite a challenge. But I think we're very, very fortunate that globally we have fantastic registries. And as long as we facilitate some sort of exchange between our registries, we should still be able to make this happen and have people sort of residing wherever they want. Nicole, you mentioned this, what can I do next and am I eligible for future trials? And I think this is a really big thing. The sort of things that may be of concern could be, for example, contamination of long-term follow-up data by a new experimental treatment. That could be any experimental treatment. It wouldn't have to be another genetic therapy. There's a possibility that a new sponsor could be concerned about inheriting a previous sponsor's risk. Seroconversion, as we've talked about, or some sort of antibody response to a prior gene therapy could lead to either blunted efficacy or an enhanced inflammatory response. And of course, there's going to be ability to access some sort of data from another sponsor if that happens. So I can see all of these could be concerns, but I think there are ways to mitigate a lot of this. So first of all, if we come back to the geography and the logistics, it may be possible to allow non-trial sites to provide the long-term follow-up services or shared care models so that people can have those long-term follow-up tests, which are usually a lot less specialized than the original trial, and that they may be able to have that at somewhere much more close to where they actually live. I mentioned the mention of the remote and digital technology in the EMA document and the registry data, which I think is really important. In terms of these concerns about inheriting a risk, I think it is undoubtedly possible for people to look at tissue and figure out which of various gene therapy products might have contributed to any signal of adverse event that is seen. We need just to be prepared to do that. And I am a bit of an optimist and perhaps not very realistic, but some sort of shared data set approach to this along the lines of what you were talking. If we are putting the person with CF really central and foremost, this has got to be something that we think about as we move forward. So I'm going to come on to something that's very dear to my heart now, and this is progressing some of these therapies into the pediatric age range. In terms of the rationale, of course, it is totally logical. CF is a life course disease. The defect is there at the time the baby's born. In fact, it's there in utero. We know that from certain organ damage. Symptoms start early, it is progressive, there ultimately are components of organ damage which are irreversible. So we could end up treating too late, and we've certainly seen that from CFTR modulators. So the modulators have really told us that there is, for some of these problems, there is a window of opportunity. We may even be seeing in some contexts prevention rather than trying to reverse disease. And within the context of modulators, there's been regulatory support for very rapid progression down the age range, very quickly. So the first thing to say was that the adult phase 3 trials all included adolescents, and then quite quickly we went down in age so that we now have ivacaftor license for 1-year-olds, and we're doing clinical trials of Kaftrio or Trikafta in 1 to 2-year-olds already. And the way those have been allowed to be designed are really based on looking for safety, looking for PK, drug concentration, and very fortunately having the sweat chloride readout as a pharmacodynamic readout, and then extrapolating safety for some of the youngest children. And that's meant that many of these trials have been single arm. They haven't had a placebo arm. They've been much easier to recruit to, and in some ways they've been quicker. I think there are downsides to not having a placebo arm as well, but we probably won't go into those now. There's also the ethical imperative for children not to be ignored in clinical trial design. So those are all the rationales for potentially going down into age range. And of course, it's already happened. This is a really great review, if anybody wants to have a look at it, of where gene therapy had got to by last year. And you can see that actually very, very early on, 1990, the first child with severe combined immune deficiency at the age of 4 was treated with gene therapy. And there are now multiple licensed treatments, all of the ones in yellow and in green here, for children or for children and adults. So the field's moving pretty fast. This table also came from the same paper. And if we have a look at what the indications are, what are the diseases that these children have licensed genetic therapies for? There's a few things that they really feature, and that is that they are very severe diseases, even in the early stages. They're largely life-threatening in the short term, and many of them are progressive and irreversible, and many of them have no other treatment alternatives. So that's where the field is at the moment. So perhaps we could consider that when thinking about children with cystic fibrosis. And certainly I think most of the pediatricians in the room would agree that most young children with CF are not critically ill and have a very short life expectancy, at least at the moment. The rate of progression is slower than for many of those diseases I showed here. And for many of these patients, there are alternatives to slow disease. They're not correcting the basic defect, but they do at least slow the disease progression. I think in this context that unlike many of the other pediatric genetic therapy trials, good safety and efficacy data from older populations is totally mandatory before we take this into the pediatric population for cystic fibrosis, my personal view. However, there is a possibility that children could actually benefit more than adults from some of these approaches, particularly because of the inhalational route. So they've got better lung health, they may get better delivery more homogeneously out throughout the airway, and they may therefore have better efficacy. But one of the challenges we've been facing over the last 10 years or so is how to assess that efficacy. I suppose the first question is, do we need to? Regulators have said we don't need to for many of the modulator trials. I think that we should, and I think that we should so that we can best balance against the risks, which there may be some, but also so we can support prescribing and reimbursement decisions. Somebody's already talked about cost of this, and we know about the sorts of cost conversations that are happening globally for modulators. I think not having efficacy data in children would be a big problem. But for that, we do need more sensitive pulmonary outcome measures than we're using in adult trials. And we've made really good progress with multi-breath washout and lung clearance index to the extent that is now the primary outcome in the vast majority of CFTR modulator trials that are done in children. And I think that that's something that may be considered to support this. But we're also looking at novel forms of imaging, and I'd like to draw people's attention to the very enhanced sensitivity of some of the MRI imaging techniques, which actually use contrast. In this, it's oxygen-enhanced MRI. Chris Short is a PhD fellow working with me and will be presenting some of these data tomorrow. And you can see that these types of techniques really have some enhanced sensitivity compared with what we're doing at the moment. I think we need to think more carefully as well about ways of adapting our trial protocols for children. It's not good enough to just say, oh, we need 2 mils of blood because that's what the lab was validated for. We really need to get our labs validating on much, much smaller blood volumes, finger pricks, etc., and think more about this. And then the other things, I think, things like contraception, consent, etc., they're not specific to genetic therapies, but some of these genetic therapies potentially going to last an awful lot longer. So we may need to think about this much more carefully in the younger children who are going to grow into adolescents or even young adults during their periods of follow-up. So just to slightly paraphrase this Shakespeare quote from Romeo and Juliet, I think we need, in this context, to go slowly enough that we can make sure we're being wise. And, you know, I'm all for including children in clinical trials, and I've been very involved in this field in the last few years. In this context, I think we really, really need to think carefully and be wise before we rush headlong into what we can do. So thank you.
So then our last speaker in this rockstar lineup is Dr. Damian Downey, who is professor of respiratory medicine in the Wellcome Wolfson Institute for Experimental Medicine at Queen's University in Belfast. He's a CF physician at the Ireland Adult Centre, and he is also the immediate past chair of the European Clinical Trials Network.
Great. Well, thank you very much for the introduction, Jen, and the invite from Jen and Nicole to speak to you today. So these are my disclosures. And this is a slight outline of the plan for today's talk. I'm going to go through some of the regulatory approval for CFTR modulators. Nothing new there, of course. But I'm going to tie in then about the CFTR variants, access to modulators, the eligible population, and importantly, the non-eligible population, and some data on expansion of access, and then a slightly more aerial view on health inequalities, and then where do we go? So this is the information on the modulators and the regulatory approval. This is obviously data on Trikafta in the US, Kaftrio in Europe and elsewhere, and otherwise known as ETI. So this is the FDA in the US, indicated for those with at least one 508 mutation or with a rare variant with in vitro evidence of response. And as you know, that's the 177 rare variants. It differs a little in the European Union, the EU, with EMA, is indicated for those with at least one 508 del mutation. And that's on the basis of the need for clinical data rather than just reliant on the in vitro data. The regulator now in the UK, the MHRA, and this is indicated for those with 508 or with a minimal function mutation or homozygote for 508 del. And that may be the indication, but reimbursement can be slightly different in using either the European EMA or the FDA license. And then in Australia, indicated for those with at least one 508 mutation. So I bring this to your attention because this is really a focus on the 508 because then this will show some European and some slightly more global data on that. So this is information which you will be all very familiar with. This is from the CFF patient registry, the data report of 2022. And you can see there, I'd say that on the 508, just at the top, percentage of individuals of roughly 85%. And obviously reciprocally at the bottom with neither 508 or unknown at 14.6%. So the data is pretty clear, and it's the 80 to 90% potential access to this drug makes it quite simple. But it is not quite simple because things vary across different countries, and therefore the eligibility for drug. It's quite a busy slide, apologies. This is data from the European registry, again from the same year, 2022. This is the most up-to-date available data. And just to orientate you, the different countries there, and on the dark blue, sorry, it doesn't show terribly well. The dark blue are those that are homozygote for 508. The lighter blue is heterozygote, and then the light blue with no 508. So just concentrating on the 508. And you can see just at the bottom with the dark blue and the lighter blue, roughly 80% will have at least one copy. So not too dissimilar. But if you look at the individual countries, you can see there's a significant difference. At the top, there's really quite low prevalence of 508, but the data on some of the countries is quite difficult to recover, and therefore one can't be totally reliant on that. But in some of the countries, particularly in southern Europe, there is 60–70% or so would have 508. So again, different. And if you talk about access from a European perspective, for example, with one 508, it's not 80–90%. In some countries, it is much less. And this is just a graphical representation of that. The darker blue is the higher percentage of 508, and the lighter blue. And you can see that will have less 508. And there's a concentration of that in southern Europe and more in the eastern part of Europe. And then this is access to ETI in Europe. Now this data is a little bit old because it's 2022 data. The pink is on the basis of license and reimbursement for over 6 years of age at that time. Of course, things are different. And then the lighter purple was a license and reimbursement for over 12. So you have the license, but reimbursement was a little bit different for different countries because reimbursement was country-based as opposed to Europe in total at the one time. And it was slightly piecemeal as well. And I'll draw your attention to the green, and that is at that time with no access, which is still maintained in quite a few places in Eastern Europe. But does that matter? Well, there's some modeling data to suggest that a delay could have health implications. So it's not everything at the one time. There was differences in reimbursement and differences in the age group of reimbursement, and you can see that has potential implications for people down the line. So we've already seen a little bit of this. Scott showed a different representation of this, and I just want to talk a little bit about the eligible population in the US. Again, this is data from the 2022 registry report, and just draw your eyes to the right part of that bar chart, where you can see ETI in green, this is access or actually prescribed, and then the other drugs in different colors. But it's that yellow population at the bottom is a sizable proportion of the community that may be eligible, but not prescribed. And this is a little bit of data, or some of the pros there, and this is pulled again from the registry report, but also with some recent publications there in the ERJ in pediatric pulmonology this year, showing what about that cohort, some lower median sweat, some differences in health insurance, and not only in the cohort with 508, but also in those rare variants, lower care utilization. And the report has talked about quite complex factors, but also race. You can see there on one of the more recent publications that racially and ethnically minoritized people with CF are less likely to be prescribed ETI. So you can see this 90% with the potential access to CFTR modulators. When you start cutting into it in different countries and at different times and different accesses, things do change. What about the non-eligibility of CFTR modulators in the UK? And this is obviously UK data from 2019-2020 report, and this was published a couple of years ago. And this was stratified by people with CF with at least one copy of 508. 91% with at least one copy, 8.6 obviously with no 508. Now, I'll just bring your eyes just over to the table and the overall percentage. 93.4 of people in the registry of white with at least one copy of 508, 96%, but with no 508, that relative and that pro rata percentage drops to 70%. And I've just pulled some data on the Asian community with 2.6% over, or 2.8% overall, but then with at least one 508, that's 1.2%. But really with no 508, you can see that's almost 20%, a fifth of the population with no 508. And that has significant implications, which I will get on to. And similarly, this is non-eligibility and similar data for CFTR modulators in the US. And again, there's a disproportionate number of people with CF who are ineligible for modulators are members of racially or ethnically minoritized groups. This is data from the 2022. Again, 95% of those CFTR prescribed from white and eligible, again, 70% in the UK, 76% in the US. And subsequently with non-white, 5% prescribed, but in the ineligible population, it's a quarter. So there's a significant differences. And that's really important. These are a couple of papers. The first one on your left, published a couple of years ago. This is a really important paper from Megan McGarry. And this is prior to CFTR modulators. Again, racial and ethnic minorities had increased morbidity and mortality from CF compared to non-Hispanic white patients. And this is a quote from the paper that disparity in access to CFTR modulators in racial and ethnic minority patients will negatively impact health inequalities in CF. And so there's quite challenging data from quite a simple, this is 90% for everybody has access to this and that's okay. It's not 90%. And when you do have access to it, or it may not be prescribed, and the challenges of having more challenging health prior to the commencement or the potential commencement of that really does have implications. And this was talked a little bit about as well. On the right-hand side is a paper from just last year. This is a single-center paper looking at those taking part in clinical trials at a single CF center, that a smaller proportion of those people, again from a minoritized racial or ethnic group, participated in a clinical trial as compared to people identifying as non-Hispanic white. So less within the clinical trials, and I think this is a really important part for the CF community to understand. So moving slightly to expanded access, and I think this is a really important paper from the French CF clinical trial network led by Pierre-Regis Bourgel. And this is with people without a 508 CFTR variant. There's been several publications on this over the last number of years. The first compassionate program was launched 2.5 years ago. And this is the recent publication just last month in Lancet Respiratory, where now 479 people with CF have received ETI for 4 to 6 weeks. So 83%, 83 had at least one FDA-approved variant. And you can see then it was 98% responded and continued. You can see the increase in FEV1 as one would expect. But importantly, there was 360 with no FDA-approved variant and no previous modulator use, and about half of those responded. And I think the really important message here is not relying totally on in vitro testing to predict clinical response. And this is the power of networks, where you can pull a lot of these data together, and the proposal here is creating an international database that assembles all these individual reports and case series. And this is really important to have that clinical data to pursue license extension. So I'm going to zoom out a little further and look at health inequalities. I've hopefully tried to demonstrate there's quite a lot of challenging data on not only access, but potentially response and health inequalities in the Western world. I'm going to zoom out a little bit and look at this from a global perspective. And this are some sobering points of that a quarter of the population worldwide have no or inadequate access to essential medicines. Now, we know that health outcomes and survival in CF have improved immensely over the last 20 years, but this is predominantly in high-income countries. Less well-defined, certainly, on the poor outcomes remain for people with CF in the low-income and middle-income countries, and I'm going to show you some data on this. And this is quite a statement of providing optimal care for all people with CF will be really challenging on a global scale. So back to Europe, and this is a change in FEV1 over the years from 2011 to 2021. And you can see, if you just take age 30, you can see the average FEV1 and the increase over time due to improved care, CFTR modulators and so forth. It really is quite dramatic. But again, it's not the same across all countries within Europe. And from that same paper, they look at high-income countries on the right-hand side, and on the left-hand side is the lower-income countries. So if I'll just bring you to the one on the right-hand side, there's three lines, the blue, the yellow, and the red. The blue is data from 2011. Yellow 2016 and 2021. So for any age group, again, you can see the improvement in FEV1. If you move across to the left and look at the same data for the same age group, you can't really see dramatic changes in FEV1. So you'll see the disparity again on the basis of the income of the country. And the age groups as well, you can see them tailing off quite dramatically. So we've already seen some of these slides today, and I'm just going to go through some of the data and this. And you'll have seen this paper, I'm sure, many times. This is now 2 years old, and this is looking at the number of people diagnosed with CF. So I'm going into the people who have been diagnosed with CF, the access, the non-access to modulators, but then we come into the cohort of people who have not even been diagnosed. So this is your diagnosis, and you can see the higher, darker colors are the number of patients. You can see data from the US, Europe, and so forth. The gray areas where there's no information available. And I particularly want to draw your eyes to India. This isn't working terribly well. Of 600 people diagnosed, in Algeria 180, and we'll come back to some of those, some of that data shortly. So this paper's been updated and just published not too long ago in Pediatric Pulmonology. And this is the estimated, and of course there's multiple challenges with estimated data, we know, but if we can use it as some form of direction, the number of people with CF estimated to be undiagnosed. So we're now in a different population. And you can see even across Europe, the US, there's a significant percentage of people that we believe to be undiagnosed. But I'll draw your eyes back to the India and the Indian subcontinent on some modeling data, and this is looking at the prevalence of the mutation in populations, migrant populations in Europe, and you can and the US, that you can potentially predict what the numbers would likely be. So 600 diagnosed, but predicted to be around 45,000 undiagnosed, which is more that's in the registry of the US. So the challenges become even greater. And I've just highlighted a paper that's just been published a couple of months ago looking at the diagnosis and treatment of CF and the major challenges in such a huge 1.4 billion people in the Indian subcontinent and the challenges that they face, and is really worth quite a sobering read. So I'd mentioned Algeria and India, and these are just, again, just picked a couple of papers. The one on the left in pediatric pulmonology just this year was just looking at a case series of 34 pediatric cases, retrospective study, 34 children, significant diagnostic delays. And a point I'll come back to about limited therapeutics is a cost a quarter of the minimum wage, with 9% who died at an average age of 9. And this is, on the right, is the predictors of mortality in children with CF in India. And this is a classic Kaplan-Meier survival on male and female. But the reason I put this graph up is because the x-axis is measured in days, not years. So what are the challenges in low and middle income countries? And this is from Scott's paper in Lancet Respiratory a few years ago now. Challenges obviously with the diagnosis, the basic diagnostic tests and lack of availability, and of course the impact of illness, of dehydration, diarrhea and so forth, and sweat chlorides really unknown. Newborn screening, again, this is the challenge with this. If there's a relatively low population incidence, then it's not really a priority for that healthcare system. And of course, the organisation of care is difficult because specialised centres, MDTs, and so forth that we take for granted in the Western world are within the confines of a very disconnected, at times, health service with very much different funding models in potentially different parts of the country. Challenges with training, and I've pointed out the medicines of 20 to 60% of health spending when it's usually in the teens that you would expect in the Western world. So it's the largest family expenditure after food. So the costs are quite huge, both in monetary terms, pro rata relative terms, but also in the impact of healthcare. And then clinical trial infrastructure is limited and industry partners are, I guess, less likely to engage. So the last couple of slides. So how do we move forward? And there has been, we've alluded to this over the last talks about advocacy, not only to expand diagnostics, but also to expand care in the low and medium income countries, cooperation with governments and stakeholders of CF care, support training and mentoring of teams, and this is certainly happening across Europe with partnering with established CF centres to those less established and developing CF centres as a system within Europe, education for families. And these are really important comments that have come through about equitable access to therapies that have achieved regulatory approval, and to address the global disparities in modulator access. So if, just to highlight, I started from the 90%, which that percent drops dramatically when you look at the undiagnosed population. So this is just to circle back on my last slide over the talks and to pull a few things together. So the development of new CFTR-directed therapies, and I use that as a umbrella term, requires us to increase engagement of the CF community in research trials, expand clinical trials in people with CF who identify as members of racially or ethnically minoritized groups, and extend clinical trials in areas or country where the population have mutations that are not eligible for mutations, because that differs in different countries. And I showed you some of that data maybe in Southern Europe and Eastern Europe and so forth. And that's the importance of clinical trial networks, is to acknowledge that, to see that, and try to have some corrective action. And this is all about building partnerships, again, as well, with the low, medium income countries. And of course, we do need to acknowledge the fiscal challenge of that restricts global access to CFTR modulators. So these are some big hitting statements. Not all the answers are there, but engagement is key and discussion is the way forward. Thank you very much.
I think we'll ask all of our speakers to come up if they're willing for a panel discussion.
So if you're in the room, if you could just come to the mic and introduce yourself and then ask your question. And then we also have some questions that are online. Do you want to start with the online ones, Nicole?
Sure. Well, one is an announcement from Recode, actually, that they have enrolled their first participant. So exciting news. And actually, one I think I might direct to you, Jen. The 4DMT data looks quite promising. However, what do you expect in terms of longevity, given dilution of DNA with epithelial cell turnover? We will not hit a wall with inability to redose.
It's a great question, even though we didn't actually talk about it a lot today. And Scott, you're welcome to jump in if you'd like to. But so the question is about redosing of AAV-based therapies. So one thing I'd point out from our data in his short time, Scott didn't have time to show, but I showed at the European meeting, and I think I showed last year at NACFC, is that there were people who were in the study who had pre-existing antibodies to the capsid, and they still had CFTR expression on biopsy, regardless of that pre-existing antibody presence. The other issue is we don't really know yet. No one's actually shown before now that you can actually get your vector into the lower airways, so we don't know how long it's going to last. Presumably, if you get it into enough basal cells, it'll be a lot longer than if you're just getting it into the respiratory epithelium. One of the things that we're considering is whether or not we should be rebiopsying patients perhaps, or participants perhaps at a year or 18 months to see if we still see expression at that point. But right now, it's sort of a black box. I think you've heard from all of our speakers that we talk about the path to the cure here in the US, but really it's a winding road that's going to continue to change and evolve as we learn new things about all of these therapies. Any other questions online?
No more questions online, but I actually had a question for Steph, actually. You had mentioned just the importance of geography in selecting a clinical trial site. And for these genetic-based therapies, not every trial will be offered at a single site. So how do you balance that selection then and trying to match clinical site with the trial that you might be interested in?
To be honest, like I said before, there are parts of the country that I actively avoid research opportunities in. And so I'll kind of look at other areas that are having these studies, these trials. And to be honest, I pick based on where I feel like I'll be the safest, or what area sounds the most fun, to be totally honest.
That's a great answer.
Patrick?
Hi there, Patrick Harrison, Cincinnati Children's. I think Jane said that we should consider shifting the gene therapies from adults to pediatrics at some point in the future, if it's safe and efficacious. But what if over the next 5 years we find it safe, but not efficacious? Under what situation would you say it's worth the risk to evaluate in younger people? As Jane mentioned, they're generally healthier, better lung function. I'm just wondering what the longer term plan is if we don't, which I would love that we get a result, but if we don't, what are the options?
Is this question for me or for Jane? Jane, do you want to go first?
I think I understood what you asked. Did you mean if we find it safe but not efficacious in older patients, would we still take it into pediatrics? Well, I think I would want to answer a lot more questions from the population who had been treated before trying it out in them. So I don't think there's anything magical about childhood. It's to do with disease progression, isn't it? So some of the adult population who would have enrolled in the trials that for the group perhaps didn't show an efficacy signal, some of those adults might have had earlier stage lung disease, and you might want to look at them as a subgroup and say, is this something to do with degree of lung damage, access to the epithelial surface, or something else? I think if you did all of that and you couldn't find a signal of efficacy anywhere, I would be astonished if somebody then wanted to take that into pediatrics without a really plausible explanation of why it should work in them when it didn't work in adults. And I'd be also quite surprised if regulatory would let you. I don't know if others have got a different view. That's a bit black and white, I know, but...
I guess I would ask what you mean by efficacy. So if someone hasn't had IVs in 5 years, is that efficacy, even if their FEV1 didn't improve? Because that might, I think, push people a little bit differently than if there was absolutely no signal in any of the measures that we use. But I agree with you that if there wasn't any signal in any place, I wouldn't sign my child up for a gene therapy when you're not sure what's going to happen down the line in terms of participating in other trials and redosing and all of the things. I don't know, Rena, do you have any thought about that? Would you enroll your child in a study if there hadn't been any evidence that it worked yet?
Probably not, especially because I didn't mention, but my 4-year-old, he actually has autism too. So he has a dual condition that we're managing. So I would definitely want to know if it's going to work ahead of time or if there is a possibility that it would work before enrolling my 4-year-old.
I can hear you. Can you all hear it? Maybe try flipping it on.
Thanks. Oh, and just to clarify, I think this question is mostly for Steph and Rena, if that's okay. So as we think about pushing genetic therapies forward, specifically gene editing therapies, one of the challenges is kind of classical animal models, right? And so a lot of the ways that we, target these therapies is to specific places in the human genome. And the human genome is different than the mouse genome or the ferret genome. And so I was just wondering how you thought about safety and efficacy in that context. So for example, if there was a potential drug, a potential therapy that we showed could work on a human genome and human cells in the lab, and we could show that it was safe and, we were able to deliver that therapy in animals, but we weren't able to show, phenotypic reversal or targeting of that specific, therapy in an animal model because the animal didn't have a human genome. How would you think about some of those considerations in terms of safety and efficacy in animal models?
So let me just rephrase it. So thank you. If a drug actually they thought worked in an animal model or a cell model, but they actually hadn't, so the CF mouse doesn't actually get CF in their lungs, for example. So they showed it was okay in the mouse, but they hadn't actually proved that it worked. Would an animal model be enough for you to say, sure, try it on me? I think is your question.
Okay. So I just want to drive home just how desperate I am for any options at all. I'll try anything. So yes, absolutely, I would do it. No question.
So for me, if it doesn't have, like the animal doesn't have the genome, for cystic fibrosis, they don't get it. So I don't know, I'm a little bit on the fence, especially with the toddler, just because he has autism as well. If it's not shown to be effective, then I wouldn't consider enrolling him. But if it is scientifically, if it is a high percentage that it would be effective, then I would consider enrolling the 4-year-old.
Thank you. Terry?
Hi, Terry Laguna from Seattle Children's Hospital. I want to bring this back to Steph. So first, I want to just say thank you for your bravery in essentially coming out to a room full of people that you just met. I know that isn't easy, and you talked a lot about how your feeling of safety, both with a part of the country, but also with the research team. And you're talking to a bunch of people who do research and are part of research teams. So if you're comfortable, would you be willing to give us some pieces of advice on how we could make the environment more inclusive, more welcoming? This morning there was a session on LGBTQIA individuals who responded to a survey telling providers how you felt. And so with a room full of people that are listening now, would you be comfortable telling us what would be helpful to create a more inclusive environment?
So as far as the perspective from a person who's LGBTQIA, this is not a one-size-fits-all sort of answer, so I'll preface it with that. But while I was participating in research recently, I traveled to a new clinic and was very apprehensive about meeting the new team. Upon arriving, I was checking in at the pulmonary research clinic, and everybody at the desk was wearing rainbow lanyards, which immediately put me at ease. There are some other great ways, rainbow stickers, pronoun stickers or pins, and even if your pronouns are, you feel like it's easy to assume your pronouns, me seeing that pin allows me to relax and say like, okay, this is a safe person. Because if I had gone into that situation and not felt safe or felt like maybe somebody was bringing their biases to that appointment, I would have been out of there. And I probably wouldn't have participated in any more research because again, that was my first time participating in research. So creating a safe and inclusive environment for everyone is essential. Absolutely. Thank you for asking that question.
Thanks.
Kathy, I'll get you to you in just a second. Rena, I know how your late diagnosis of your son happened the second time around, but could you explain to everybody else who doesn't know the story so they understand a little bit about what you said about mistrust?
Sure. So for my oldest son, he's 23. So in Louisiana, we're from New Orleans. We didn't have the newborn screen there for him. But for the 4-year-old, we knew that it was a possibility that he would have cystic fibrosis. And when we moved to Georgia from Hurricane Katrina in New Orleans, we evacuated. And the pediatrician who was for the 23-year-old was also for my baby. So I went and I made him aware, like, hey, I'm pregnant and I want you to be my baby's pediatrician. And will you take us on? And he was all for it. And I did everything that I could to make him aware that there's a possibility that this child may have cystic fibrosis. Fast forward to getting genetic counseling, going to the hospital, planned C-section. I made all the staff aware of, hey, there's a possibility that the baby may have cystic fibrosis. Could you please newborn screen him and let me know of the results? They said I had to wait for them or get back with the pediatrician. So I went back to the pediatrician. It's 2 weeks, we're out the hospital, and I'm licking my baby. Like, he's salty. No matter how much I bathe him, he's severely salty all the time, and I'm second guessing myself. I'm in a house crying. I have friends and relatives coming over, and they're also licking my baby. So everyone is licking the baby, and some days they're like, well, he doesn't taste salty today, and I'm like, well, he does taste salty. So I bring him back to the doctor, and I say, hey, the baby is really salty. Could you please check on his newborn screen results? And he printed them out, and he said, see, Mom, you should be happy. He passed the newborn screen. And while the news was, I guess, happy news, I just knew in my heart of heart that something was wrong with my baby. So tears were streaming down my face, and, the pediatrician, he goes, well, you're paranoid, because your son has because Gerard has cystic fibrosis. And so I just looked at him, and about 2 weeks passed, and I said, well, gosh, what could I do? So I called back to the Children's Healthcare of Atlanta, and I knew the pediatric team there, and there was a nurse practitioner. And I said, Bree, I had a baby, and I believe the baby has cystic fibrosis. What can I do? And she said, Rena, if you believe your baby has cystic fibrosis, there's no one else who will know that. So I believe you, your baby has cystic fibrosis. So she said, go back to your doctor and let them know that we recommend all full siblings receive a a sweat test. So I did that, and I went back to the doctor, and he was still saying that I was paranoid. He reluctantly sent the test off to Children's for my baby. I brought him, and his first test was inconclusive, and we had to wait 21 days for retesting. That is going to change in the future in my state, where you don't have to wait 21 days. I got that information yesterday from Dr. Linneman. But yeah, the pediatrician had to call me back and tell me my baby actually has cystic fibrosis. And so I was really relieved at that point and very happy to know that I wasn't going crazy and that he could get the enzymes that he so needed, because his symptoms was he was constantly hungry. He would cry for 12 to 14 hours every single day. And I just remember thinking, like, this is repetitive of what I went through with the first child. It took me 4 years to get him diagnosed, the first one.
So yeah, I just want to, she pointed out Tuskegee earlier in her talk, but it isn't just Tuskegee, it's ongoing behavior in medical systems that impacts people's desire to participate in research trials.
Yes, because after having an experience like that, I'm kind of reluctant because my concerns, they really fell on deaf ears. Like, I remember calling the hospital back and raising my concern with them, and I was just rerouted back to the pediatrician, and it was just like a circle and a loophole, rabbit hole I went down.
Thanks for sharing that, Rena. Kathy?
Hi, Kathy Ramos from the University of Washington. I want to say thank you to everyone for speaking, and especially to both of you for sharing your personal stories. The reason I got it was more for a comment to say that we need to keep in mind the power dynamic that exists when Steph was saying, I'm desperate and I would do anything. We have to be careful how we talk about these trials and enrollments, because it's a huge decision, and we do this every day, and it's a new topic for you, and it's life-changing potentially if you go into a trial. And so it just reminded me that that power dynamic is huge with any patient population, but especially with populations that have had this history and ongoing episodes of injustice that keep happening. So it's something we all have to keep in mind.
Yes, agree. Go ahead.
Thanks so much for your perspectives. Along those lines, I'm kind of curious about, I think there's a number of people on the panel that would have an interesting perspective on this, but like in patients with, or in families with multiple people affected with CF, how does that affect like the algebra that goes into those shots on goal and preclude people, like do siblings develop antibodies to one therapy, for example? And how do we answer those questions?
Yeah, I mean, I can go first. I don't know if you want to jump in, Jane. But I think, it's a great question. Again, I'm going to say that we don't have a pathway. We have this winding road because we don't know things like that. I mean, presumably, if you haven't actually been exposed to it, you wouldn't. But for AAV, it's everywhere. So you may have pre-existing antibodies. I mean, I know for those of you who read Isabel and Anna's Stern's book, they talked about during the Dornase alpha trials how they sat next to each other when they were randomized, and both of them were inhaling both things just because they were worried about the other person. Hopefully that's not happening in these trials, but I don't think that we know yet the answer to that question. Jane, Scott, did you want to say something?
Yeah, I wanted to ask Steph a question. It came up before about out-of-pocket expenses, and we were talking about this earlier in the week. Can you just tell us a bit about what the magnitude, how you managed that, and what the clinic did to respond?
Yeah, so out-of-pocket costs were a big concern for me as somebody who needed to travel for research. I wanted to make sure as many of my needs were taken care of on the front end before I traveled. And then, of course, with these types of trials, you've got some longer visits and things that are reimbursed, like potentially Ubers or food or whatever, that really adds up for somebody like me. You think, if your per diem is $50 to $75 a day, that's a lot. I get paid $1,200 a month from the government. And so it really adds up for me. But one thing that the clinic that I worked with did, and the sponsor, was as soon as I got those reimbursements sent to them, the research coordinator had them submitted within the day, usually, and the sponsor paid out within 24 to 48 hours. So it made it so much more manageable for me as a patient, especially on longer visits. That was essential and something that is not always the case, because I have participated in research where that doesn't happen as quickly.
Lynn?
Hi, Lynn Quattel. I'm the Cystic Fibrosis Data Safety Monitoring Board. This was a great session. Thank you. And Nicole, you definitely touched on this, and everybody else touched on it a little bit. But I think that one of the million-dollar questions is, how are we really going to execute long-term follow-up trials? And we've talked about this a lot. And everybody has these guidelines, and the FDA has guidelines. But if you look at the FDA's guidelines, it's like semi-ridiculous, because they really just put the onus on a care center to do, on a physician to do it. And it's not even the care centers necessarily. It's unrealistic to assume that sponsors will really do that. It's not even in their budgets necessarily. And sponsors, as we all know, don't always have long lives, and they disappear. So I think that one of the things that we really have to address is, how are we truly going to do long-term follow-up, and who's going to be responsible for executing those?
I think that's a good point. And I wanted to kind of follow up on a comment Jane meant too about the potential for more centralized long-term safety data collection. And, do you have any ideas of how we can approach regulators on this type of supportive idea? I mean, in some sense, there are already surveillance programs for even, that are more government-backed for some of these programs. So you can even think about outside of CF, that for cell and gene-based therapies, this may need to be a broader initiative for the field as a whole to really progress this and tackle some of those long-term safety issues if we're going to do it right and with these challenges in mind.
I think it's a really great challenge, and I think it's really important. And actually, I hadn't even thought of the point that you just made about sponsors falling by the wayside. That, of course, we've seen it, haven't we? Many of the small molecule companies that were here 10 years ago are not here now. I think the minute you start talking about all diseases outside CF as well, my very small brain shuts down, because suddenly that becomes a problem that's almost too big for me to think about. I think we're really, really fortunate in CF that we have the registries. And the European registry, at least, has got a license for post-marketing surveillance. Maybe we could convert some of those tasks into being the long-term follow-up of trials. That would obviously require buy-in from the companies who are doing the studies.
Yes, I mean, the issue right now, though, I think that the registry is an amazing tool, and we've talked about this. But right now, the way the registries exist is they do not capture the information that we need for long-term follow-up. So we need to figure out, again, how to pivot that so that it does, and again, who's going to pay for that?
I agree with you, but I think those things can be changed within registries, and probably doing that is a much easier and more streamlined task than setting up a whole new system, I would have thought.
Yeah.
And who's going to pay for it? I actually think the sponsor of the trials should be the person who pays for that. And that might mean that different people are having, you're having your data collected through that long-term follow-up, and someone else is having it collected through that. But how straightforward that's going to be, I don't know. Do you have a better solution?
Oh, no. Well, we have talked about the DSMB actually creating almost an ancillary DSMB, so that we would at least we would execute them. But again, the issue is, how do you formalize that? How do you make sure that the money is in place to execute these studies? But potentially, we would create sort of an ancillary DSMB that would help do that.
Scott, go ahead.
So, a question for Nicole. What's happening in the discussions with the regulators about this point, and with the companies that the trial centres are considering?
So what's exciting, at least with the U.S. regulators, is that there's a number of new initiatives in the rare disease field. The Accelerating Rare Disease Cures Program, there's an innovation hub that now is meant to span both CBER and CDER. There's different divisions within the FDA, and these NABTs span both of them. So, we've been working with regulators at I would say, the very senior leadership level to raise the attention of these issues. But start with basic education on how the CF landscape has changed dramatically and how our trial design landscape needs to change, but also starting to bring up some of these innovative design concepts and that we are going to have to do things differently than our traditional precedent design path. There's an extreme amount of enthusiasm, and I'll just say on behalf of Dr. J.P. Clancy, the regulatory and policy team at the CFF, is that we do have a pathway for continued engagement with the agency. There's a lot of enthusiasm about the support of external controls, given that they know we have a limited size patient population, and that particularly in the cell and gene therapy space, that this could diminish over time with exposure, depending on how this pipeline evolves.
All right, well, being mindful of the fact that Joe Paluski is about to teach us a lot in the plenary, I don't want to be late for that. So hopefully you guys have seen there's still a lot of questions. We've made a huge difference in the lives of a lot of people, but the gaps in health equity have gotten even bigger for people who already had those gaps in the first place. So as you're listening over the next couple of days, just keep that in mind as we're trying to move things forward so all people with CF have a really good therapy. Thanks.